So for passive transport, we've talked about simple diffusion, simply just going from higher concentration to lower concentration, molecules diffusing. We've talked about facilitated diffusion, which requires proteins, either channel proteins or carrier proteins. And now we have one more category of passive transport that is very important to understand, and that is osmosis. Osmosis is going to be movement of water across the membrane. Sometimes we can't move molecules. Sometimes we need to move water to make concentrations equal. And this goes back to the whole concept of solute concentration. Remember, a solution is a solute plus a solvent. Remember, the solvent is the liquid portion, in this case water, and the solute is going to be what is dissolved in that water. Solute concentration is going to be very important in this story, and remember, we express that using molarity. So if something is a 10 molar concentration, that's 10 times greater than a 1 molar concentration. It's stronger, it's saltier, it's more sugary. So this is how we're going to express how water moves, because we're looking at solute concentration. Sometimes that solute can't readily move, but if we can move water, we can balance that solute concentration. So let's look at a couple of examples of that. And there are going to be some important terms that apply. I need to erase all of this to make room. So we're talking about osmosis, water movement. And let's look at a couple of different scenarios with a cell. So here's a cell. I'm going to, just for the sake of example, use sodium chloride as our solute. Because we've talked about sodium chloride, it's a term you're familiar with, you know it's a salt. So let's pretend that inside the cell we have a 1 molar sodium chloride concentration, and outside the cell we have a 5 molar sodium chloride concentration. Where is the solute concentration higher? Inside the cell or outside the cell? It's much higher outside the cell. And in that case, water is going to tend to move toward wherever the solute concentration is higher, to try to dilute that solute concentration and make them equal inside the cell and outside the cell. So if we're trying to dilute where it's higher, do you think water is going to tend to move into the cell or out of the cell? It's going to tend to move out of the cell. It's going to move toward where the solute concentration is higher. So water is going to move out of the cell. We call this a hypertonic environment for this cell. Hyper means above. If you're hyperactive, it means your activity level is above normal. Hypertonic, we are talking about tonicity, which relates to solute concentration. This is a hypertonic environment. It is more salty outside the cell. And in this situation, water tends to move out of the cell. It's going to move toward this higher salt concentration to try to dilute it. So rather than just moving the salt in and out, we can move water, and that is called osmosis. So in this hypertonic environment, water tends to move out of the cell. And over time, whether this is a plant cell or an animal cell, what do you think is going to happen to that cell if water keeps moving out? It's going to shrink. It's going to shrivel up. So over time, both plant and animal cells will shrink in a hypertonic environment. Let's look at the opposite scenario. Let's look at this situation where the solute concentration outside the cell is actually lower or below what's inside the cell. So I'm going to use those same numbers. I'm just going to reverse them. So let's pretend that inside the cell we have a 5 molar sodium chloride concentration, and outside the cell we have a 1 molar sodium chloride concentration. Remember, water is going to move toward the higher solute concentration. So will water move into the cell or out of the cell? It's going to move into the cell. Water is going to tend to move into the cell. We call this a hypotonic environment for the cell. Hypo means below. If you have hypothermia, it means your body temperature is below normal. Hypotonic, we're referring to the environment of the cell. So we're saying the solute concentration outside the cell is lower than inside the cell. And in this case, water tends to move into the cell. So water moves into the cell. If this is an animal cell, over time, it's going to get so big that it could actually burst. If there's a big enough solute concentration difference that enough water moves into the cell, that cell can burst. So animal cell can burst. A plant cell actually purposely sets up this scenario, because if it concentrates solutes inside its tissues, that means when water is available, it's going to get pulled into those cells. So if you've ever had a sad, dehydrated houseplant, and then you add water, and a few hours later he's all perky again, that is a happy plant cell. They have concentrated solutes in their cell, so when water is available, it gets pulled in. So water moves into the cell. An animal cell can burst over time, but a plant cell, because that plant cell has a cell wall on the outside, and I won't draw the whole thing, but and it should be green, cell wall on plants, that plant cell will not burst. That plant cell is actually going to become happy. It wants to be full of water, and it's what's called turgid. So plant cells, over time, in this hypotonic environment, where the solute concentration is lower outside the cell, plant cells become turgid, T-U-R-G-I-D. These are happy cells. Obviously not literally, but it's a good thing in plants, bad thing in animals. So here, plant and animal cell did the exact same thing. They both shrank. Here, animal cell is going to burst, plant cell will become turgid. In the middle of this, we have what's called isotonic. If something's isotonic, iso means same. That means solute concentration inside and outside are the same. And in an isotonic situation, water is going to move into the cell and out of the cell at an equal rate. So isotonic is same solute concentration inside and outside the cell. And water movement is going to be in both directions. This is what the cell is trying to achieve. It's trying to achieve equilibrium. Sometimes you can't move the solute, so you have to move the water. This picture shows that. It shows that there's a higher concentration of sugar on one side versus the other. The sugar is more concentrated here. The sugar can't move across this selectively permeable membrane, but water can. So we're going to shift more water this direction to balance that out. Dilute it so that the concentration is the same. If I gave you 2 beakers, and in one of them I put 1 gram of sodium chloride, and in one of them I put 5 grams of sodium chloride. And I said, I want these to taste equally as salty. I want some salt water, and I want them to be the same saltiness. Which one would you have to add more water to get it to be the same saltiness? You would have to add probably 5 times as much water to this one to get it the same saltiness. So that's what we're referring to with this water movement. Osmosis. This is very relevant in living systems. It has to do with water balance. And if you take physiology, you will learn more about water balance in human systems. This water balance is a big deal. It's called osmoregulation. Regulating water in the body. Just one more picture of this same story. So hypotonic, remember, that means the environment outside is a lower concentration of solute. So water is going to tend to move into the cell. Plant cells become turgid. That's a normal, happy cell. But the animal cell is going to burst. Isotonic, this is normal. We're going to get an equal movement of water into the cell and outside the cell. And then finally, higher concentration of solute outside the cell, water is going to move out of the cell, and that cell is going to shrink. Now in a plant cell, this cell wall stays rigid, but what's going to happen is that plasma membrane is going to shrink down as the water leaves. Same for this sad little shriveled cell. Okay, so that is osmosis. Also considered passive transport, no energy required. Next, we're going to start talking about active transport where energy is required. We've covered passive transport, no energy required. Remember, passive transport is always going from high concentration to lower concentration. So no energy is required. That's not always the case though. Sometimes we need to pump things to an area or move things to an area where it's already high because we need more of it. Or sometimes we need to get things out of the cell. There's already a lot of it outside the cell, but we need to get rid of all of it or most of it. And that's called active transport. Active transport does require energy in the form of ATP. And it is going against a concentration gradient. This is going uphill, upstream. This is going from area of low concentration to higher concentration. So this is when you already have very little on one side of the membrane and you're going to get it to where it's already high. A very good example of that, you'll learn about in more detail if you take physiology, is called the sodium-potassium pump. There's a specific pump, it's an enzyme in the cell membrane that pumps sodium ions out of the cell and potassium ions into the cell against a concentration gradient. So what that's saying, in just very simple terms, if it's pumping sodium out of the cell against a concentration gradient, there's already a lot of sodium ions out here, outside the cell. If we just had one more left, and we wanted to get it out of the cell, that is going to require energy, because that is going from low to high. And conversely, for potassium, what if we already had a lot of potassium in the cell, and we want to get this last one in? Again, that's going to be against a concentration gradient, and that is going to require energy to cause that enzyme to work. This is just another schematic of that same thing. We're pumping sodium out, and we're bringing potassium in. Again, you'll learn about this in more detail. What you need to know at this point is that it's called active transport. It's against a concentration gradient, so it does require energy in the form of ATP. This is going to be very important also when you understand membrane potential. And that's a very good example of active transport. Nerve impulses, everything you do requires a nerve impulse. And membrane potential is very important to understand. There are going to be times where we need to move ions in and out of a cell to create a net electrical charge on one side of the membrane. So remember, ions are either positive, which are the cations, or negative, which are the anions. And moving those ions around can change the net charge either inside the cell or outside the cell. In this situation, this is showing a net positive charge on one side of the membrane and a net negative charge on the other side of the membrane. This next example is a little more clear. You can see on this side we have more anions. Remember, anions are negative. And because we have more of these negatively charged ions on this side of the membrane, it's going to have a net negative charge on this side. The opposite is true on the other side of the membrane. We have more cations, more positively charged ions on that side. So this creates a net electrical charge on one side or the other, and that is called membrane potential. That is going to be very important to understand if you take physiology. Also anatomy. So this is just another way of showing that same thing. Positive on one side of the membrane, negative on the other, and we can change that. We can move those sodium ions in and change that negative environment to being more positive, changing that membrane potential. So just a quick review of the two categories, active versus passive transport. Remember, passive transport involves just simple diffusion, high to low. It's not going to require any energy. Facilitated diffusion is high to low also, but we either have a channel protein or a carrier protein because we're talking about molecules that can't just move across that phospholipid bilayer, in part because of these hydrophobic tails. And then we have active transport. ATP is required because we're going from low to high. You can see 1, 2, 3, 4, 5 of these diamond-shaped molecules already in the cell, but we want to get the rest of them in. That is going to require energy. So passive versus active transport. How things move across cell membranes. Okay, then we have this major category of movement. This is called bulk movement. This is endocytosis and exocytosis. And endocytosis is going to really fall into two categories. So endocytosis is going to be moving materials into the cell. So bulk movement, we're not talking just O2 and CO2 at this point. We're talking about things that are much bigger than O2 and CO2. Endocytosis is going to be how we move bulk materials into the cell. So endo, into the cell. And exocytosis is going to be how we move bigger materials out of the cell. Exo, exit. Endocytosis is going to have two categories. One is going to be for small little liquid droplets, almost like cellular drinking, and then the other is going to be cellular eating. So under the category of endocytosis, we have two important terms. Pinocytosis. This is cellular drinking. This is small fluid droplets that come in. On the other hand, phagocytosis is going to be how certain single-celled organisms such as protists actually eat. This is going to be how our white blood cells engulf foreign particles. Bacteria, viruses, dust, pollen, whatever foreign particles get into our body that the white blood cells perceive as foreign, there are certain phagocytic white blood cells that are going to devour that material. So phagocytosis This is cellular eating. And we have phagocytic white blood cells. These phagocytic white blood cells carry out phagocytosis. They wrap materials in membrane, sometimes entire cells, and they bring that in and digest it. So phagocytosis, pinocytosis. Two forms of bulk movement of materials into the cell through endocytosis. Wrap it in plasma membrane, bring it in. Sometimes we have what's called receptor-mediated endocytosis, where something has to bind with a receptor on the cell surface in order to be recognized and brought in. So sometimes that endocytosis can actually be what we call receptor-mediated. This is how we process bad cholesterol. So receptor-mediated endocytosis. It involves molecules actually binding to a receptor, and then we bring them in and process those materials. Exocytosis, again, is bulk materials moving out of the cell. We saw this when we were looking at the endomembrane system. If something is wrapped in membrane through a transport vesicle, see this is a phospholipid bilayer carrying a protein product, probably made on the rough ER, probably processed at some point in the Golgi. And now it reaches the plasma membrane, those membranes fuse, and those protein products leave the cell. That is called exocytosis. Both of these involve wrapping something in membrane and then moving it either into the cell or out of the cell. Endocytosis and exocytosis. Okay, a couple of quick review questions about transport across the plasma membrane. If an animal cell is placed in distilled water, what will happen to the cell over time? Well, this can be kind of a tricky question. You have to think about what is in distilled water. And remember, this all goes back to something called solute concentration. You need to think about where the solute concentration is highest. Are there any solutes in distilled water? No. If it's truly distilled water, it should just be H2O. Nothing else, no solutes. So that would be the lowest possible solute concentration. So if a cell, a living cell, is placed in distilled water, do you think a living cell has just water in it? Or do you think a living cell probably has some solutes dissolved in it? Different ions, different sugars, whatever the case might be, there are solutes in a living cell. If your cell is the same solute concentration as distilled water, you've got a problem going on. That is not normal. So that would be called a hypotonic environment. Remember hypo and hypertonic? That distilled water would be hypotonic relative to the cell. So is water going to move into that cell or out of that cell? Over time, water is going to move toward where the solute concentration is higher. In this case, it's going to move into the cell. So over time, in that hypotonic environment, water will move into the cell. And remember, a plant cell becomes happy, turgid, an animal cell will burst over time. So it will lyse or burst. What will happen to a plant cell in a hypertonic solution? So that's the opposite. That's where the solute concentration is higher outside the cell. And remember, water is going to move toward where solute concentration is higher. Water is going to move out of the cell, and in this case, both plant and animal cells will react the same. They will both shrink. So the cell will shrink or shrivel over time. What are the major differences between passive transport and active transport? You should really be thinking of two major differences. One involves energy, and one is concentration gradient. So remember, passive transport, no energy required. Active transport requires energy. And that's pretty easy to remember because it's kind of in the term. Why does passive transport not require energy? Because it's movement of molecules along a concentration gradient from high to low. Active transport, different situation. We're going from low to high. You're going to have to pump it. You're going to have to use energy to get those materials to move. So, concentration gradient and energy are the two things you should be considering.